Am I audible? Anyone please confirm? Am I audible to you guys? Okay, thank you, Chandana. Yeah, I guess we can start. In the meanwhile, people can join if they want to. So, I know Mansi. Chandana, if it is fine with you, you can unmute and just tell some basic information about you, from where you are, what you are doing right now.
No, ma'am. Actually, I'm traveling.
Okay, no problem. So, can you just give a brief introduction about yourself? Is this your first class with me?
No, ma'am. Last week I was attended.
Okay,
This is
okay,
my second.
no problem. You can unmute yourself. Sorry, you can mute yourself. Yeah. So, I guess we can start with today's session. And as always, I hope you guys enjoyed this week's sessions. You learned something new. So, anyone wanna give a try what you guys learned in week 4? A very brief, like pointer wise summary of what you guys have learned. If you are okay, just raise your hand and I'll call out your name. And then we'll discuss one very interesting topic in brief, and then we'll go back to our questions uh, to which might help you guys in solving this current week 4 lecture assignments. Week's assignment, I'm sorry. Yeah. So just a request to all of you who are new here, who are joining. Just raise your hand. I just want to know your very brief introduction. What is your name, from where you are, guys, what you guys are pursuing, just so that we can make this session interactive, and you guys feel free to ask questions. Just raise your hand. I'll ask what you want to know, and I'll try my level best to sort your doubts. So, anyone in the meeting right now who is joining session for the first time with me for this particular course? Anyone? If yes, just raise your hand. Okay, seems like all of you have taken the session with me at least once. So you guys know the drill, right? So, okay, who gonna brief me what we learned this week? There were, I guess, 5 sessions, right? Yeah, there were 5 sessions. So what we learned, what you guys remember, okay, just don't go with the chronology order. What you guys remember from week 4? Anyone volunteer? I'm not gonna cut your marks, I'm sure. So don't worry. Even if it is wrong, we can discuss. Yeah, so go ahead. Anyone? We learned about neurons or something, right? Skeletal, muscular junction neurons. We gradually switched from integumentary to skeletal to muscle to now neuron. You can see, right? We are moving from outside to inside gradually. I hope you guys are able to make clear mind maps of these layers which we are following throughout the week's lecture. So yeah, so what we learned in week 4. How many of you have completed week 4 lectures? Just raise your hand. Okay. How many of you have partially completed week 4? Not complete, but like some of the videos you have gone through for week 4. So none of you have seen at least a single video also from week 4. Okay. So I want to suggest you guys, I have already given this, I guess, last week also. Without going through the week's sessions, you might not be able to get the maximum effect of these live sessions, right? Because I'm not covering anything new. And even in all the live sessions, I guess we have 3 live sessions for this particular course. So in all the live sessions, we don't discuss anything new. We don't teach you guys anything in the live sessions. We just try to solve your doubts. So doubts will come when you will see the week's sessions and videos and read. And also we try to solve previous year assignment questions so that you guys might have some idea what to expect in the current week assignment as well as in the exam, so that you can solve for yourself. So for that purpose, this input is needed from you guys, that you should at least watch the week's lecture. Not fully, it's okay. I understand that every one of you is involved in one or another thing. I agree. I also came from that background only. I understand completely, but uh, I will still request you guys to try to up to date with the week. At least more than 50% you should be covering before coming to live sessions. I'm not saying that you cannot come. Obviously, you can come. You will learn, definitely you will learn something if we are spending 2 hours together, right? But you can get more benefit if you guys have some prior information, at least topics in mind, what was discussed in the week session, right? So this will be helpful for you as well as for me to uh, like what to say, comprehend topics, because I don't know what you guys know and what you guys don't know. So our basis is basically week 4 session. So whatever the depth and whatever the topics were covered in week 4 for this particular day, I'm saying, so that will be the thing I will assume that you guys are aware of, right? And I cannot go again and again on the very basic topics, though I don't mind going into it. If the topic is very important and people are having trouble in it, I will definitely try to make sure that you guys are able to understand so that we can move ahead together. But again, just try to follow up with the week's lecture. Mansi, you can put your hands down. And if you want to give us a summary, if possible, or if you are interested, you can go ahead. This might help other people also, that what was covered in the current week, this week 4 lecture. I guess you are the only one who actually went through the lectures. So do you want to give it a try?
Yes, ma'am, sure. So good evening, everyone. Ma'am, I am audible.
Yeah, good.
So this week is dedicated towards muscles and neural system, and the interlink between these two. The major topics that we covered this week are the length tension relationship, sliding filament theory, excitation contraction coupling. Um, so here we studied, sir taught us about the muscle and the force generation, in which the arrangement of myosin and actin filament is responsible. Then we are headed towards a fast twitch and slow twitch fibers. Um, and then sir also taught us about the latent period, and then the graph between the fast twitch and the slow twitch fibers, which is dependent upon force and time.
Yeah.
Then we were taught about the force velocity characteristic, in which there is a relationship between velocity and force and weight. Then in the lecture we sir taught us about extrafusal fibers and intrafusal fibers, and the correlation between these two. And extrafusal fibers are those fibers which sense the outer environment, and intrafusal muscles are those muscles which can be linked to the neural system, um, which senses the external environment and give the information to our CNS, which includes brain and the spinal cord. Um, then there is a flow chart between the muscle, sensors, and the CPU, uh, and the diagram of the extrafusal fiber. Then in the last two lectures, we have been taught about nervous system anatomy and signaling, basic technologies of neural system anatomy, circuits, electrical signals involved in this process, and correlating them with mechanical movements and information processing, such as memory, awake, and sleep. And yeah, this was
This
about.
was, yeah. Great. Good going, Mansi. So I hope that you guys at least know what topics were covered in this week. So as you can see, it's a very wide variety of topics, right? And we cannot cover everything again and again. The lectures are already at your doorsteps, literally in front of your screen. So you can always go back and look at them and learn new things. So what I want to discuss with you guys, which I guess has discussed in the week lecture, but I want you guys to think more on it and we can have active discussion about it. So in a hope that you will get deeper understanding as well as proper imagination and mind map of things, how actually we are sensing our environment, right? So every living organism, right? Even a non-living thing, we all sense our environment, external environment, right? So can you give me an example of a non-living thing sensing a external environment? Anyone? Any vague idea? Any vague idea? Nothing is wrong here. Anyone want to give it a try? I'll give you a hint. What happens during rainy season, especially in dry states or northern part of India, when rains are a particular like season, right? We have a dry heat and then we have uh, rainy season. So the moisture is high in the environment. So mostly the wooden doors and those things, they get certain characteristics, right? That phenomena is adsorption, the surface phenomena. The water gets onto the surface of these non-living things and they become very hard to open and close, right? It's a very common phenomena to be observed, especially during rainy seasons in the northern part. So though directly they are not sensing, the wood is not saying, okay, come from the environment, the water molecule can stuck to it, right? It is not a active process. So, but in a way, we can still see that there is a change in a commodity when the surrounding change or the environment change. So we can say that is sensing somewhat, not obviously not absolutely. And every living thing obviously have senses. They respond to different stimuli, right? For example, living organisms start their day from sunlight stimulus. Plant grow towards sunlight. Plant grow towards gravity, right? The roots grow towards gravity, should grow against the gravity. And what other stimuli we have? Food or chemical particles is a good stimuli, right? Even a microorganism, for example, bacteria, fungi, they all show a peculiar movement and motion with respect to the presence of food. I don't know whether you guys are aware of it or not, but yes, Mansi, do you have a question?
I know, ma'am, I want to add
Acha.
something in. Ma'am, like for example, phototropism, geotropism.
Yeah. Those all are the, I was just telling the name of the stimuli. For example, photo, gravity. The tropism means movement. So when the stimuli is present and the movement occurs, that phenomena is called the name of the stimuli and the tropism word. Okay, so if the motion is because of the light, it is called phototropism. If the motion is because of gravity sensing, it is geotropism. Great. So there are, for example, there have been researches already. Everyone has seen E. coli, at least not in the lab, but on social media or such places, right? E. coli is a bacteria, right? It has a flagella associated with it. So it has been observed that the way the E. coli moves in its surrounding, it changes based upon the chemical signals from the food source. So what I mean by this is for example, this my room, your room, just imagine whichever room you are in right now. It's a 3D space, right? So if a microbe is present, for example, on the floor, or you have a water droplet on your floor and the E. coli is present there. So in that water droplet, the E. coli is constantly moving. They are moving in random direction, just like Brownian motion. Like I hope that everyone is aware of Brownian motion, the random motion of the particles, right? So they have a very random motion without any direction, without any motivation. They are just randomly swimming here and there. But the moment they sense the chemical signal from the food, for example, I have dropped a glucose molecule, because glucose is the primary source, right? Most of the living organisms are carbon based. So if I put a glucose molecule or something like that, it has been observed in the Petri plate that that random motion of the E. coli becomes more oriented and directed motion instead of being randomly here and there all the time. If the food source is there, due to the chemical signaling and the sensing in the microorganism, they start their motion in a directed way towards the food source. Okay, so imagine a cell which we cannot even see, a very primitive cell. It's a bacteria, it's a prokaryotic cell, right? A very primitive cell. Even a primitive cell has the mechanism of sensing, right? A very, very basic phenomena which all living organisms display. Correct? Then this is about prokaryotic organism, that the moment they got a stimuli or a sense or a change in the environment, their behavior, their action, their way of processing things, because their genome will also change, right? Now they need to process the food. For processing food, you need enzymes. For enzymes, you need proteins which make those enzymes. For proteins, you need mRNA. For mRNA, you need transcription, translation, right? For that, you need DNA replication, and all those stuff needs to happen in that moment. So all these changes just because there is a sensing mechanism associated with the living organism. Correct? Now imagine about the complex organisms like us. We constantly sense multiple things simultaneously all at once. You agree with me? If you guys agree, just give a thumbs up. What I'm saying is that we always sense 24/7, we sense different things all at once without even realizing that we are sensing and we are in connection with the environment. How many of you agree with this statement? Just give a thumbs up. None of you. Okay. Why you don't think that we sense 24/7? Anyone want to give it a try? If you guys are not giving me thumbs up, which means your views are contradictory to mine, right? So I want to know what are the opposite views. So now you guys need to tell me why you think whatever I'm saying is not correct. Narendra, you want to try? Kumar, do you want to give it a try? That whatever I'm saying, is it right or wrong? Even right now, you guys are sitting, right? So I hope that you might be sitting in a room which has ceiling fan and that fan is on. So you can feel air on your skin, right? You can feel air flowing on you, right? how you know that you're feeling it? What is the thing which is making you feel that the air is flowing around you? It's a way of sensing, right? And if you're not sitting in a room with a fan turned on, then you might be sitting at a place where there is other auditory signals. Maybe there's road outside, vehicles are going on, maybe pets are around you, they are barking, or there is light there's high light, there's low light, depending upon like wherever you are sitting. So these all are senses, correct? How can you sense whether outside is dark or light? It depends upon your sensory organs, your senses, right? So 24/7, even when we are asleep, right? Even when we are asleep, if something drops or a dog barks or a vehicle literally blows horn near to our ear, we get up, right? So even when we are asleep, our auditory system is functioning, correct? We are sensing. So by saying that we sense 24/7, even when we are asleep, is because of the sensory mechanisms, sensory system in place, that we are constantly in connection with the environment. You guys are sitting, right? You might feel uncomfortable after a certain amount of time if you are sitting idle for hours. So who is sensing that? If that sensing mechanism is not there, what do you think will happen? I'll give you a try. I'll give you a thought experiment. Imagine if the candle is there and you're putting your finger to the flame. So if you guys are not able to sense the heat from the fire, you won't remove your hand, correct? You are removing hand because you're feeling burnt. What the feeling is sensing, correct? So if you don't have a sense or feeling of your surrounding, what will happen? So if your finger is in a flame and you are not feeling the heat from it, you won't remove your finger. And then at the end, what will happen? You will be burnt, and burnt to the extent without any scope of recovery, right? Because you won't remove your hand. Similarly, something is hot, something is cold, something is spiky, something is like gnarly. So all these things assure our safety, because if we are not able to sense, we won't take action to make sure that we are in our optimal condition. Everything comes back to our first lecture of this course. Every living organism want to be in the optimal condition. Those optimal condition not necessarily only depend upon the temperature, pressure, oxygen concentration. No, those are our daily to daily interactions with the environment also. If the seat is not comfortable, you wanna change that. If you're not having good sleep, you try something else to get good sleep. If, for example, the food is not tasting good, you take actions against it, right? So if you don't have all these feelings, all these senses of your surrounding, you won't take any action to make sure that you are at your optimum, correct? Till here, whoever is with me, give a thumbs up, because I need to know that you guys are active in the class. Everyone, please respond. And if you're not able to understand, let me know so that I might simplify things. If I'm keep talking, it makes no sense, right? You guys should understand things also. Okay. So by saying this, we assured that senses are important part of our living being, of our living system, correct? Now coming to us, we humans, we have very complex sensory mechanisms, right? Even other animals have more complex sensory mechanism than us, but we are the ones who are able to consciously think about it and study ourselves, right? So talking about sensory system, everything on our skin, this is the outermost layer, right? So you are literally separated from this environment and your internal body is literally separated from your skin. So what do you guys think? Our skin is highly sensitive or is not sensitive at all, according to your basic understanding also, whatever we have discussed? So what do you think? Our skin should have high sensory processing abilities, or it should not have sensory processing abilities? Yes. More responses in the chat box. Guys, you need to be active in the discussions. Chandana, Kumar, Narendra. We are very few today in the class, so I can actually pinpoint you guys. So try to be open. Yeah, please be active in the discussion. It helps me also to like take my lecture forward at a pace in which you guys are able to understand, right? So our skin has to be sensitive. Now, at the molecular level, right? At the molecular level, what are the things which are sensing? Correct? Because from outside, if we talk about this fire example, what we sensed the heat, right? But that heat was on our skin. Who told us that this heat is bad for us and remove your hand? Take a decision about it, right? If I change the conditions completely, for example, if we go to winters, in winters, we love to hold warm things, right? We love to wear gloves. We love to wear tea, hot beverages, and keep them very close to our hand. We don't remove our hand. But if I ask you guys to hold a steel glass, which has literally fresh tea poured in it, you won't touch it, right? Because it will be too hot for you guys. But the same scenario, same condition will change if I talk about the same conditions in winter weather. Correct? So who is telling what to take the decision in the current frame of things? Right? So that thing indicating that there has to be a connection between the external environment, the sensory system, and the decision-making system. Everyone here with me till now? Give a thumbs up. Give a thumbs up if you guys are understanding. Okay. So what I just stated, I said to make decisions based upon the situations and the stimuli and the sensors and the sensory system, there has to be a channel for a stimuli to interact with the system, which senses things, and to the control system, which makes decision, right? So by this, what I mean the sensory system, the sensory things, which I'm going to discuss in the coming few minutes. The sensory system, our brain, correct? Now, if I'm sensing something on my foot, my brain is not in my foot, right? My brain is sitting right here, indicating that there has to be pathways or connections which connects my foot sensory system to my brain's decision system. Correct? Just like roadways. If tomatoes are grown at certain part of India, let's say in northern part of India, tomatoes are available everywhere also, right? Why? Because of the transportation system, a very magnificent transportation system. So it is not necessary that wherever the source of a thing is, you need to have a consumption only in those areas only, right? Because we can distribute things. So similar thing is what our body is based upon. We have sensory system throughout our body. Those are connected to our brain via connections. Those connections are neurons, right? So we sense something anywhere in our body, that information is quickly transferred to our brain. The brain analyzes the situation, it takes decision, and again the decision is sent back to the localized environment from wherever the stimulus was there. Again, it depends upon what kind of stimulus and what kind of response you want. If the response is of, for example, it's a very point source, it's if it's a needle and you are picking your finger through it, the response will be to keep your finger out. So it's a localized response. The response is only for our left hand's index finger, correct? The response is not for our left leg also that move backwards, correct? But what if it's a flight or fright situation? You need to literally run, correct? Then it is not a point source response. It's a whole body response. Your whole body needs to get into the position of running if there's a threat around, if that is the response from the stimulus, correct? So the location of the response can be localized as well as distributed. This will be decided by brain. The point which I want you guys to understand very clearly, and I hope that I might be able to make sure that you are understanding the concepts as well. Things which happened on my foot traveled to my brain via neurons, analyzed, traveled back to the localized environment via neurons, and then the response was taken care of. What exactly is traveling? The stimuli is not traveling. If the fire is there, the fire is not traveling through my neurons, right? Or for example, if there is water and you don't want to step on it because you don't know how deep the puddle is, correct? The water is not transporting inside you, metaphorically speaking, for this particular situation, right? So what exactly is the thing that is converting a stimuli into a thing which can be processed by a control system and which is able to travel from neurons? You guys are here with me till now? Give a thumbs up. Okay. Am I not clear enough? Okay, I have few responses. Okay. Yeah. So what exactly is the thing that is traveling? The stimulus is not traveling, right? The neuron itself is not traveling. If it happens, okay, what do you guys think if this is the scenario that something I put my foot on, it's a sharp object and I need to remove my foot. So this whole process will take multiple steps. The step will be my foot being on a spiky thing. Me now internally, what will happen? Something will sense it, it will transport it, it will take a decision, then the signal will again come to my foot and then I'll remove it. Okay? What is traveling? If I say now, and if you guys don't know, and I'm just saying, for example, if we are reading it for the first time. So if I'm saying, okay, the neuron which was present at that part of my body, which sensed something, Mansi, I'll get back to you in a minute, huh? Which that part of the body, and the neuron is present there. We have already made sure that the neuron is there to sense things and transfer things. So if I'm saying that that particular neuron will travel back to a brain, give the information to a control system, then again go back, or say a second neuron is going back for relaying the signal. So do you think it's a convenient design? Yes, Mansi, I think you want to answer this question only. Yeah, go ahead.
As you said, not the neurons travel, nor the nor the stimulus travel. I think it is the neurotransmitters which travels across this.
No, but I want to know, see, if you don't know anything, because we are not mugging things up here, right? We are understanding concepts. what I am saying, if a neuron travels from the stimuli source, from the point source to our brain, and then go back again to the point, to the responsive site, what is wrong in this? It can be done, right? What problem it will have if a neuron is transporting from one place to another place, or traveling from one place to another place for the signaling purposes? What are the drawbacks? Like,
Then
what
it
do you
would
think will
be
happen?
then it would be a very complicated process.
Complicated in what sense?
Mom, you are saying that neurons, if neurons will move from one place to another place, then which difficulties would be facing, right?
Yeah. You said because it will be a complex process. So give me one complexity which you think will arise because of this system. Whatever you can think of, it's completely fine. It's a hypothetical question anyways.
There will be no proper channels. It
Okay.
would be a hotspot situation.
Okay. Okay, let me guide you through it. It's okay, you can put your hands down. One basic thing, me saying this, that I stepped on a spiky thing, the signal traveled, the decision was taken, the response signal was sent to my foot, and I quickly removed my foot. All this thing happened at what time duration? In seconds, right? The moment you touch a hot thing, you quickly remove it. Or you just keep it there, you wait for your brain to process it, and then you will remove it. Doesn't happen like this. Correct? It's a very quick response and very immediate response. So if a neuron is supposed to be traveling from the point source to the control center, don't you think there will be a huge latent period? Obviously, because by the time you are putting your foot on a spiky thing, your neuron is traveling. Don't you think in this meantime, chances are you might actually get pierced by that spiky thing? Because you haven't made decision yet. There will be a huge latency. There will be a huge time difference between the stimuli and the response. But in real life, there is no such thing, right? We quickly do actions depending upon our stimuli, right? So this is very basic thing which you can imagine. If a same thing happens when we are I guess you guys might be aware of conduction, convection, and radiation, right? Modes of heat transfer. So in conduction, convection, radiation, there is a presence of whether the particle is moving, the energy is moving, or nothing is moving at all, right? Or the medium is required, not required, and all those things. I'm not going to go deep into it, but it's the same phenomena. If you want one source particle to move from one end to the entire length of the other opposite end of a thing, there will be a latency period, there will be a delay. But in real life, we don't see such things. In disease conditions, we do, but in normal conditions, we don't. That is why we can completely neglect this possibility that a neuron will travel from the point source to the responsive center of our brain, right? I hope I'm able to convince you guys. If you guys are convinced with me, give a thumbs up. Okay. Still few people are not convinced. I hope that you might ask me questions, maybe if you're comfortable enough. And yeah. So we can completely neglect the occurrence of stimuli traveling through us, neurons traveling from stimuli point to the responsive center. So what else is there which can travel, right? Coming back to our this week lecture, we studied, and even in the previous lecture, when we were discussing about that we are electrical beings. You guys remember that discussion we discussed, right? That we have charge distribution, correct? Charge distribution, gradients, electricity, electric potential, all these come into play the moment you have some charged entities around you, or within you, or with you, right? If a charged particle is present, for example, there are few charged particles. I'm not saying whether they are positive or negative. We don't know what characteristics they have, right? So if charged particles are present, what kind of, I don't want to use the term force because then you guys might not answer it. But if charged particle is present, what things you can expect from a charged particle present in a vicinity? Just fly with your imaginations. Imagine a chlorine or a potassium or a sodium. It's literally sitting in front of you, in a beaker or in front of you, wherever you can imagine. So what are the things that charged particle can do or will happen to it? Because it depends upon the conditions also. But what will happen if a charged particle is present in front of you? Very, very lame answer. Any lame answer, everything is welcomed here. Just give it a try. What do you think will happen if a charged particle is present in front of you, or wherever you want to imagine it? And I'm not saying what kind of a charged particle is there. I'm
Flow
just
of
saying that it is a
electricity.
charged particle.
A flow of electricity.
Flow of electricity, we can say if we know there are distribution of opposite charges, but we are not saying that. I'm just saying it is a possibility. I'm not denying it because I haven't given you any particular condition. Imagine where I want you guys to think and also develop this ability to start thinking in imaginary way. Then only you will be able to learn maximum things, right? So if I asked you question, there's a charged particle. Now you can take any hypothetical charged particle of whatever you like or whatever you want to give it a try. So for example, I'm taking a potassium ion. Potassium ion is a positively charged ion. It's a positive charge. Positive charge indicates lack of electron in it, right? Because the electron was not there, hence it is positively charged. So its inherent quality will be, as soon as it will be able to get an electron, it will keep searching for it, right? The moment, because it is deficient in it. A potassium ion is what? It is deficient in electron. So its existence is there because the electron is not present there. So how the ion want to be? What is the optimum condition for the ion also? It is when it is completely filled with its electron, right? With the required electrons. Hence, the whole sole motive of the potassium ion will be to get the electron by any means. Correct? That any means, it depends upon what element you are talking about. Then we can go into physical chemistry, but we are not going into it. So if a potassium ion is there, it's an ion, it lacks electron, and it wants electron to be complete and to be optimal and to be best self, right? So what it will do, it will move here and there. If it is moving here and there, it is moving in search of what? Electron. And again, I said there were charges, right? So I have taken one potassium ion. There can be another, let's say, any negative ion is there, is sitting right here. So if this potassium is moving very fast randomly here and there, as soon as it senses a negative cloud, because there's a chloride ion is negative in charge. Negative charge means the negative force will be around it, the electronic cloud will be around it. So the moment it reaches towards, or at least in the vicinity of chloride ion, its direction again will become oriented and directed. It won't be anymore a random motion, right? So as soon as it reaches your chloride ion, what it wanted electron. What chloride has electron. What chloride wants now, keep the potassium aside, what chloride wants? Chloride has excess of electron. It doesn't want to be in that state, it just want to give it, right? So whoever comes to him, he will give the electron, right? Because he wants to be in the best possible optimum condition it can be. Correct? So when they both come closer together, the bond will form, the bond will be formed. But in the meanwhile, what happened? What the charges were doing? The charges were generating force. The charges were sensing force. I have taken oppositely charged particles, hence they were experiencing attractive force. If I would have taken same charged particles, they would have been experiencing repulsive force. Right? So whenever we are electrical beings, we have charged particles. So all these phenomena, which I hope that you guys are able to imagine things and understand the basic flow of things. Same condition is present inside us also, right? The same potassium is present inside our body. The same chloride is present inside our body. The same sodium is present. The same negatively charged proteins, positively charged ions, neutral ions, everything, every single thing you can imagine from the chemistry is present inside us, most of it, right? Not the extreme heavy elements. They are present inside us, correct? So the phenomena which we are observing here, if a positive charge is there, it lacks electron, it just wants electron. So wherever it will get electron, it will go. It will go means it wants electron, means it will go towards the positive negative charge, means it will show attractive force or attractive direction motion. If there are multiple positive ions, they don't want to be closer together with each other. They will repel each other. It will be a repulsive force. Correct? I hope that you guys are understanding. Give a thumbs up if you guys are understanding what I'm trying to say. Okay. Very few people are responding. If you guys are not able to understand, you need to ask questions, because I won't be able to know that you are not able to understand, right? I cannot read your minds, at least till today. You never know. Correct? So please be active in the class. You are also giving your 2 hours for the session. So at least try to learn as much as you can. Okay. So this is the thing. Now, coming back to the stimulus, transportation, and processing things. I got a stimulus, and that stimulus, it can be anything, right? It can be anything. The stimulus was there. I was able to sense it. I'm pressing my finger against my skin. I'm able to feel it because I know that my finger is pressing my skin. How I'm knowing this? I have charge distribution throughout my body. Correct? I have positive charged ions, I have negative charged ions, I have everything inside my body. The moment I'm putting my finger in close proximity to the other thing, the environment of this local point changed. Here, not anymore. There is not only the charge distribution of this local place, but there is force, there is pressure, there is weight, there is direction. These commodities are also here now, correct? Which means the local environment of my sensory system changed. Correct? Here comes the role of sensory molecules. If I'm saying that if I'm pressing hard, right? I'm pressing hard, which means there has to be something here which senses the pressure. This is acceptable for me. This is not painful for me. But if I put a very sharp thing here, it will be unbearable for me, right? Which means there is a limit, or there is an extent of things we can sense without experiencing pain. Correct? Yeah. So the moment I'm touching my finger to my skin here, the local environment, the local condition changed. I'm not saying what changed exactly, but something changed. Now it is not optimal. This is optimal. This is not optimal for my localized system. Everything wants to be in optimal condition. Now, if I have this finger here, which is not causing any trouble to me, am I jolting out, or am I removing my hand from the stimulus? No, right? Because it is not causing any difficulty for me. It is not causing my overall optimal condition to be changed drastically. Correct? Hence, the response is nothing. The response is, yeah, there is a finger on his head, it's okay. Correct? But if there is a knife or there's a sharp object or there's a heavy object, my response will be to remove my hand, or at least lift the heavy object as soon as possible, whichever is easier for me to do. Again, it's a very like millisecond, microsecond, we take these decisions. Correct? So the response is nothing. It doesn't mean that the signal was not processed. The signal is processed because I know that my finger is touching my skin, but there is no response. And even absence of response means there is response, right? Because the response was not to do anything. Did you guys get this point? If yes, just give a thumbs up. Otherwise, I'll clear more on this point. Okay, cool. So if I'm touching here, if my finger is placed right here, there is no response for doing anything, indicating that there is no response or no signal for my muscle, for my bicep and tricep to pull my hand down, right? The response was just to stay wherever you are. It is not a non-optimal condition. Hence, the signal was not sent to my muscles to remove my hand. The signal was just here to inform me that there is something on your hand and it is okay. You can take the decision depending upon what is there on your skin. It is my own finger, I'm fine with it. If it is something else, I might remove it, right? Again, all these things are happening at a very microsecond, millisecond level. How fast they can happen, we know this. You can experience this in your daily life. But how exactly this responsiveness is conducted, this is the beauty of biology, right? This is so fascinating. Imagine this. Just imagine you are taking response in a microseconds. But for that microsecond to happen, there are multiple steps which your system needs to do before you can do that action, right? It is fascinating, and we are not able to understand completely in everything about our system because it is so complex. There are so many variables associated with it. Coming to charge distribution and availability of small molecules, it is pretty easy. I hope that you guys agree if I say moving a heavy object is difficult and energy consuming in comparison to moving small objects and more number of small objects, right? I'll rephrase it. It is easy to move small things with A's in comparison to moving larger objects. Do you guys agree with me? Yes, no, somewhat. Okay. So coming back to the point, we have ions, we have molecules, small molecules. Here I completely am talking about charged ions. I'm talking about ions. Ions are very small, right? We cannot imagine, we cannot see even cell. Cell is made up of atoms, right? Atom has all these particles. So having a huge collection of small molecules so that we can distribute our job throughout the gradient is easier in comparison to having one thing which can transport up and down. This is how our biology and system has adapted. Instead of putting one center for things, we have distributed our controls, right? For example, if I want to remove, if I want to move my left arm, I can only move my left arm by giving instruction to my left arm muscles. I don't need to give first instruction to my abdomen. Then my abdomen is the center where all the muscle information is processed, and then it will take a decision whether to move this or not, right? It's not like this. If I want to move my left arm, I'm able to move my left arm because there's small muscles. There are muscles which are only present in my left arm, and my left arm signal is not getting linked with my right arm signal. Correct? This happens because we have distributed our roles, distributed our jobs. This distribution comes into play when, in sense of sensory organs or like transporting signals from one place to another place via small molecules. And in sensory system, we call them ions, because the moment you sense something, the neuron gets activated, the neuron changes the distribution of, or around the neuron, the distribution of ions, these small molecules changes, right? The change in the distribution of these small ions lead to the signal. That signal, which is further administered to a control center, things happen there. Again, the signal is sent back to the response center and the response is taken care of. That is why having a proper ionic balance, proper concentration of ions is needed and is important. If you remember, calcium is not only necessary for your bone health, it is also important for your muscle. And now I guess you know why. Because for signal processing, there are steps where calcium is needed for signal transferring. If the signal is not transferred properly, you won't be knowing what to do in the situation. And hence, your optimal condition will be shifted. Correct? I hope everyone is here with me till now. Yes, no, maybe. So talking about signaling, I wanted you guys to know, I hope that you guys know about action potential. If you guys know about action potential, just raise your hands. I'm not gonna ask what action potential is, just I want to know whether you are aware of action potential or not. Only one person is aware of action potential. Okay, so action potential is a way through which our signal gets, how to say this in easier terms. Okay, so how we sense things, we get to know that there has to be a distribution of ions across the neuron. And by across the neuron, I mean across the cell. By across the cell, I mean across the cell membrane, right? At the end of the day, every cell has an outer covering, which is cell membrane. Cell membrane has transporters. Cell membrane has proteins, which work as a channel between the external concentration of ions and the internal cells itself concentration ions, right? So due to the gradual opening and closing in a very systematic manner, the concentration of ions changes throughout one cell or at one cell. That further gets propagated to other cells, and hence a proper signal is transmitted. Okay, I was supposed to discuss more about it, but as you guys are not more into it, because you guys have not heard about it, so I cannot discuss it, because I have to first teach you guys, and it might take another lecture then. So I guess we can start with our questions today. And if you guys have any doubt, just feel free to raise your hand and ask. Okay. So our first question for today is, what is the range of forces generated by a power stroke in human muscle machinery? That is actin and myosin. The unit of it. Even if you don't know the exact unit, you'll get some idea from the options itself, right? Because you know what we are talking about, one muscle doing work. So what is the force generated by it? So I'll give you guys 1 minute for this question, and your time starts now. Just type your answer in the chat box. No one is on. At least give it a try, even if it is wrong. Give it a try. Anyone, give the wrong answer. Don't worry about it. Well, the correct answer is nanonewton. So if you don't even know what these units are, but if you look at the options here itself, mega means too big, right? Mega, you cannot have mega amount of it, logically make no sense, correct? Then we have Newton, then we have millinewton, makes some sense, but still millinewton and nano. If we have a smaller unit and we are talking about a power stroke, like single power stroke, so it has to be the lowest one possible among, at least for this particular question. So if you don't know the exact answer, the things are right here. And if you go back to the week's lecture, this was the place where sir has discussed what type of fiber, depending upon the length of fiber, how they generate force and what is the like range of forces generated by these muscle machinery. Okay. Our next question for today is, excitation, contraction, coupling involves interaction between first cardiac muscle and motor neurons, skeletal muscles or motor neurons, cardiac muscles or cortical neurons, or skeletal muscle or cortical neurons. I'll give you guys 1 minute for this and you can type your answer in the chat box. Your 1 minute starts now. Give it a try, even if it is wrong. Please try to attempt these questions. They will help you for your current assignment as well as prepare you for the exam if you're planning to take, which I must say that you should take the certificate. The correct answer is skeletal muscle motor neurons. And if you see here in the diagram, which I've kept here, motor neurons are cells in the brain and spinal cord that allow us to move, speak, swallow, and breathe by sending commands from the brain to the muscle that carry out these functions, right? Their nerve fibers are the longest because they need to connect from brain to our periphery, right? Their nerve fibers are the longest in the body, and a single axon can stretch from the base of the spinal cord all the way to the toes. As I already mentioned, the neurons has to be there for the communication process, right? So the question stated that excitation, contraction, coupling. So if you go back to the lecture, don't worry if you are not aware of these terms. You will get to know if you follow the lectures. In lectures, the things were pretty, in a really good manner, they were explained. And in those situations where you are not aware of these things, so we can break down the question, right? The question says, excitation, contraction, coupling involves interaction between, which means there are three things happening, right? First thing is excitation. Excitation indicates stimulus, right? It somehow says excitation is there, which means some stimulus, some change is there. Contraction indicates response, right? Because you respond on the stimuli. And coupling, which means there was a stimulus and the response was taken care of and there is a connection between them, correct? So from this way you can connect. So stimuli, it will be obviously outer muscles as skeletal muscles, as we are discussing since like past 2 weeks, skeletal muscle. And again, motor neurons, some basic information about motor and cortical neurons. Motor neurons will be connecting periphery and all those. So if you have a little bit of information, you can easily solve these questions. Okay. Our next question for today is, which of the following is or are non-excitable cell types, right? Our options are skeletal muscles, neurons, nephrons, or all of the above. It's a pretty easy question, but still I'll give you guys 1 minute for this question. Just type your answer in the chat box and then we'll discuss. And feel free to just attempt these questions. Well, the right answer is nephrons, obviously, because we know that skeletal muscle neuron, we just studied about them, right? That they are excitable, they respond to the change, correct? And nephrons, even if you're not aware of it, but I guess most of you are from bio background, so you know what nephron is. A nephron is basically the functional unit of the kidney. It is the structure that actually produces the urine in the process of removing waste and excess substances from the blood. There are about 100,000 nephrons in each human kidney. The most primitive nephron are found in the kidneys, pronephron of primitive fishes, obviously because fishes came earlier to us, right? Amphibians, larvae, and embryo of some advanced vertebrates. The nephron found in the kidney of amphibian and most fishes and in the late embryonic development of most more advanced vertebrates are only slightly more advanced in structure. The more advanced nephrons occur in the adult kidney or metanephrons of land vertebrates such as reptile, birds, and mammals. You don't need to worry much about it right now. We definitely gonna cover the kidney structure and all those things in the coming week sessions. So here I've just kept a very basic diagram just for you to see what we are talking about. And if you don't know, skeletal muscle we are aware of, neurons we are aware of, which means first option is not the right answer. Second option is definitely not the right answer because neurons are the ones which are the excitable cell types. And which means all of the above is not the right answer if first and second are not the one. So even by negative choice, we know that nephrons will be the correct answer for this particular question, right? Yeah. Coming to the next question. Our next question says what? It says, which of the following is untrue about twitch? Twitch is basically a muscle response which we have. It depends upon the duration of force which muscle fiber generate. If you have went through the lectures, it will be very clear to you guys. Okay. So the options are as follows. It is related to muscle function. It is the fundamental unit of muscle contraction. It is produced by a single action potential in a muscle fiber, or it is characterized by a short delay, a latent period, a contraction time in which the muscle tension falls to zero, and a relaxation time where tension goes to maximum. I'll give you guys 1 minute for the question. Don't worry if you don't know, just try to attempt by negative choice. Also, again, for this purpose only, I say multiple times, please go to the lectures, learn from them, and then we can have a very good discussion and we can clear our doubts more efficiently. Your 1 minute starts now. Okay. So give the answer in the chat box. Okay, good to see few answers after so long. Thank you so much for responding. Well, the correct answer is it is characterized by a short delay, a latent period, a contraction time in which the muscle tension fall to zero, and a relaxation time where tension goes to maximum. Chances are you might not be able to get the right answer if you haven't gone through the lectures. So again, I request you all to please see those lectures. And this was the part where during the session sir was explaining about the twitch force and what twitch fibers are or what twitch action is. So I'm not gonna go in detail to it, so that you guys should go back to the lecture and look into it. Okay. Our next question for today is, which of the following is a property of type 1 fiber? Right? There are different types of fiber in our body, and here we are talking about muscle fibers, okay? Because it's about muscle and movement and muscle fiber. So what are the different following is a property of a type 1 fiber? Is it they are slow twitch fibers, or they undergo anaerobic processes, or they are very easily fatigable fibers, or all of these are the property of type 1 fiber? Your time for 1 minute starts now. You can write your answer in the chat box. Even if it is wrong, feel free to drop wrong answers. Please but try to attempt these questions. They will force you to think and revisit what you know, and they will help you to make new ideas and new concepts also, so that this learning will be a long run, persistent knowledge and not just bookish information for you guys. Anyone want to try? Okay, the right answer is slow twitch, and again, it's part of the week's lecture, so you might not know it because you haven't gone through the lectures. So if you go back to the lecture of this part, you will understand what this graph means. These are the distribution. This is the time frame, and this is the force generated and force felt by the fiber. This is fast twitch and slow twitch. These were some distinguishment among fast twitch and slow twitch, and obviously you can clearly see that the slow twitch fiber is clearly written here as type 1 fiber, right? So indicating slow twitch fibers have certain characteristics, and they are also known as type 1 fibers, and there might be other names also, which you will come to know if you go through the lecture. Okay. I knowingly kept these lecture slides only so that the information and the imprinting of the knowledge gets deeper, because if you have learned from one place and if you keep rereading it, the concept becomes very strong. So I like strongly suggest to go to the lectures, learn properly, and then revisit them again. Our next question for today is, intrafusal fibers are. If you remember in the beginning of the class during the summary session, Mansi talked about intrafusal and extrafusal fibers and muscles. So even if you're not aware of them, she did mention their roles. So if you were listening to it, you might be able to attempt this question. So the question states is that intrafusal fibers are they auditory sensors? Are they photoreceptors? Are they nociceptors? The spelling is wrong, I guess. Or the proprioceptors. And obviously you have 1 minute for this, and your 1 minute starts now. Any guess, any wild guess, even if you don't know the right answer, try to break down the word. Intrafusal fibers, we are talking about motion here. What thing it will be sensing? You have 20 seconds to go. So whoever wanna give it a try, do give it a try at least. Okay, the answer is proprioceptors, which means, if you go to the lecture, you will know, but I won't repeat myself again. I hope that you guys got the strong message which I want to convey here. So intrafusal fibers are a specialized cell population in skeletal muscle found within the muscle spindle. These fibers have a mechanosensory capacity, forming part of monosynaptic stretch reflex arc. Even this was discussed. A key component responsible for proprioceptive function. Impairment of proprioception and associated dysfunction of the muscle spindle is linked with many neuromuscular diseases. Research to date has largely been undertaken in vivo or using ex vivo preparations. These studies have provided a foundation of understanding of muscle spindle physiology. However, the cellular and molecular mechanism which underpin physiological changes are yet to be fully elucidated. You can see here, right, that despite having intensive research for many years, we are aware of it. We know very few things about many things, and there are like literally a universe amount of information which is still not here with us with respect to ourselves, right? We don't understand human anatomy as we think we understand. We need more work into it. This is a very simplified representation of where exactly things happen. We have muscle, we have neurons, we have motor, especially the gamma type of axons, and there are different type of axons also. If you go back to the lecture, you will understand what these things are and how our muscles and cells are distributed and how they all are working together to ensure that we have proper and perfect response and motion at every given time. Okay, so if you remember that, not remember, but I guess you might have seen people who drink a lot, right? They start getting disbalanced. They are not able to walk straight. At the core level, these are the things, because you are impairing the neuron signaling. The moment you are impairing the neuron signaling, your movement, your motion, your organized thought, everything will alter, right? So at the base of it, it's the neuron and its signaling and the action potential and the duration of action potential, the way it is generated, how long it is being persisted, to what signal it is increased. Is it towards the slow twitch fibers or the fast twitch fibers? So these are little entities. These are the puzzle blocks, right? So at the moment you change the stimulus, the condition, or you remove one part, everything falls out of place and things become dicey. And these conditions are called disease condition most of the time. Obviously, alcohol condition is not the disease condition, but the being alcoholic is kind of diseased condition, right? Yeah. So our next question for today is intrafusal fibers, right? We just read about extrafusal fiber and indicate that they belong to similar family. So intrafusal fibers are type of skeletal muscle, motor neuron. It can be either skeletal muscle or motor neuron based on neurotransmitters or none of the above. I'll give you guys again 1 minute for this question. Even if you think you are guessing it, give it a try. Write the answer in the chat box. Your 1 minute starts now. Okay, I have, excuse me. You guys have 10 seconds. Sorry, 5 seconds. Yeah. Okay. The correct answer is skeletal muscle. Obviously, we just saw in the previous slide, as well as if you go to the lecture, this was the part where during the session it was discussed where the extrafusal fibers are present, where the intrafusal fibers are present, what extrafusal fibers are there, what intrafusal fibers are there. There was a very interesting animal example also there, which you will clearly understand if you go to the lecture, and how strong these muscles and these fibers actually play a role in your motion. Okay. So next question for today is for today's session is this: which motor neuron innervates extrafusal fibers? Right, we just saw in the previous slide that there were neurons in the extrafusal fiber, right? And then there were muscles and there were other things associated with the muscle fiber. So if you remember that slide, the answer is pretty easy. But even if you don't know, and if you know something about motor neurons as we have talked, you might be able to guess the right answer. So the question states, which motor neuron innervate extrafusal fiber? Is it alpha type of motor neuron, gamma type of motor neuron, or both alpha beta type of motor neuron, or none of the above? Yeah, both, like none of A and B. And your 1 minute starts now for this particular question. Okay, I see that people are replying. Thank you so much for that. Yeah, so even the wild guesses, even the wrong guess, just try to attempt, because while even trying to attempt, you are revisiting whatever the knowledge and information you have about these things, and it is making your concepts and learning more stronger, and also letting you know where you lag, right? So that you can go back and cover your bases depending upon your knowledge gaps. So this is how we all learn. So feel free to even give wrong answers. Don't worry about it. So you guys have 20 seconds still left. Anyone want to give it a try? Feel free to guess the answer. At the end, obviously, I'm going to give the right answer. Yeah. So the right answer is alpha type of neurons. Okay, I guess I've kept the wrong image. I'll change this image and I'll get to the correct image. I guess I've copy pasted the wrong image. It was something else. It's okay. I'll change this. Okay. We'll discuss this in the next class. Yeah, coming to the next question. If neurons were compared to wires, which we often do, and even in the session, you will see that this comparison was made, and even if it is not made, you can do it, because a wire, you can see a neuron structure we discussed. So you can make this comparison very easily. So what this question is stating, it is saying, if neurons were compared to wires, insulation can be compared to what? Insulation, you know, right? The wire is not the one which you see, the red color or pink color or blue color wire. No, the wire is actually inside that plastic coating, right? So we are asking here, a neuron is there, a wire is there. A wire also transmits electricity, right? Or signal or charge distribution. Even a neuron is doing the same thing. So what are the analogy between these two things? So if neurons were compared to wires, insulation can be compared to Schwann cells only, microglia cells only, Schwann cells and oligodendrocytes, or microglia and astrocytes. For this, I agree that we need information about the neuron structure as well as the cells associated with the neuron. But I hope that you guys have some preliminary information and knowledge about neuron. If not, don't worry, we're gonna discuss, obviously. So your 1 minute for this particular question starts now. Feel free to take wild guesses, even if you have heard something somewhere that you remember something, give it a try. Everyone should try at least. It's okay if it is wrong. It is not getting counted in your score or something. So don't worry about it. Feel free to answer. Okay, the time is up. The correct answer is Schwann cells and oligodendrocytes. So this is the very basic structure of a neuron, a very basic structure, a representative structure actually. So we have dendrites which collect information, the signals which we were talking about, right? The stimuli, the change in the distribution of ion concentration and all that. So it's been sensed by these dendrites which spread throughout our body. Then soma is the main cell body where the nucleus or the like the center of the cell exists. Then we have axon, which is the longest dendrite. It can be said as a longest dendrite, but it has special name axon, which transmits the information and the signal towards the other neuron and the other cell. Hence, the connection is there between one neuron and the other neuron. Myelin sheath is made up of like carbs, lipids, and all those things, which help in insulating the axon in somewhat. It helps in making it a faster ride, right? So we have a normal metro, right? And then we have bullet trains. They both function well enough. They will both reach to their destination, correct? But you can see the difference in the speed through which they are traveling the same distance. A metro train will travel at a normal speed, while a bullet train will travel at a really faster speed on the same conditions. So if axon is myelinated, that is a high signaling or very fast neuron. Okay, and then we have Schwann cells, which are associated with the myelin sheath and all that. And then at the nerve ending, finally, these nerve endings will be further connected to, depending upon they are motor neuron or cortical neuron, or are they connected where their shape and functionality will vary. Now, as you know, since the first week, I want you guys to know things how they actually are. And instead of imprinting images of the representative structure of cells and muscles and anything which is possible to see in real life and how they really exist, we should always have that image instead of the representative image. So this is one image of a mouse brain. So you can clearly see that these are the neurons, right? So this is the cell body, which is highly fluorescent, is dark green concentrated. And these are its dendrites and axons. Chances are we might not be able to distinguish which is the axon and which is the dendrite because we are not trained in it, but a person will definitely be able to distinguish between axon and dendrites. And also, I wanted you guys to see how neurons are actually formed. So I have this very short, I guess not even 2-minute video for you guys. Just let me share it and we'll see it. It is pretty interesting to visualize this. We don't need any sound access. It's a short video. So just let me know if you are able to see my YouTube on your screen. Give a thumbs up if you're able to see my YouTube screen. Yes, I cannot see. So you guys are not able to see my YouTube screen on your screens. Then I have to reshare it. Anyone can confirm? Narendra, can you please confirm? Can you see my YouTube screen? Just a thumbs up will work for me. Assuming that you guys can see my screen, and even if not, don't worry, the link will be there in the slides also. So if you can see the screen, just look at this time bar and look at how the neurons are growing and the network is getting more complex and complex as the time moves. It's so interesting, right? So intriguing. We are making newer and newer connections the more time is going on. Here, the time refers to what? That we are learning new things, we are getting new stimuli to make decisions about it, to respond to those stimuli. Our brain is constantly working on, right? So that constant working is in a way implemented by making new neuronal connections. By new connections, I mean the axon of one will get connected to the dendrite of other for efficient knowledge transmission from one neuron to another neuron, or signal in this case, right? This is so mesmerizing. Just imagine from where we start and where we go at the end. This is at 74 hours, 78 hours. Just look at, right? So I hope that you were able to see the video. Now you have to tell me again that are you able to see my slides or not? I hope that you guys can see my slides. So yeah, the answer was this only. And now I guess you can relate to what this still image is and what was the motion image or motion video was, right? That's how we are making connections as we are. And even when I'm talking, you are hearing, you are learning things, you are understanding things. A proper connection is taking place. Our mind is constantly working, constantly making new connections, and new things are growing in our mind, right? So last question for today is, it's a very easy question. So brain is anatomically situated dash to kidneys. It's a position respect. If you guys have went through the first week lecture, you will know what these positions are. So brain is, and even if you don't know, it's pretty easy to answer. So our brain is anatomically situated dash to, with respect to kidneys, our brain is situated where? Is it situated caudally to our kidney? Is it situated superior to our kidney? Or is it situated posterior to our kidney? Or is it situated inferior to our kidney? I don't think so that we need 1 minute for this, but still for the sake of consistency, I'll give you guys 1 minute for this question. Feel free to give answers, at least give answer for this question. I will highly appreciate. Obviously, we don't need this amount of time for this question. It is obviously superior, right? Kidney is below and brain is above superior, correct? So, and even if you guys were confused about this caudal and posterior terminologies, this is the basic anatomy, but I have also kept the structure uh, terms, structure related terms for this. So you can clearly see here the lateral view and the anterior view, the cranial view, the caudal view, the posterior dorsal view, and the anterior or ventral view. These anterior, ventral, posterior, dorsal, it depends upon for what organism we are talking about. Are we talking about bipedal animal or a quadrupedal animal like dogs or cats? They walk on 4 legs, right? So these terminology might change, but their meaning will remain the same. So I guess this is the last question. Yeah, this is the last question for today. So this is all from my side with respect to today's session. Again, please go to the lectures, because these discussion sessions won't help you to the max if you are not going through the sessions. And we genuinely want you guys to take maximum out of these courses, because you are giving your time and energy into it. So enjoy these live sessions. Try to attend as many as you can. Give answers. It will help you in understanding things. You will be able to give answer if you go to the lectures. Don't mug up everything in the last moment. I understand that these things are difficult. And also if you are not completely into it, they might seem like a chore to you also. But I can say for sure that if you're able to go through the lectures, attend live sessions after October, you will be having some great information and knowledge base for your animal physiology. And even if you're not planning to pursue it further, it's all right. But understanding human biology, understanding who you are, will at the end help in the betterment of your life and the people who are around you, right? Because everything is connected, just like neurons. So please try to follow the sessions at least halfway through it. If you're not able to complete whole sessions till the live session happen, it's all right, but at least some amount of work has to be there from your side too, so that the learning continues and there is no breakage in the learning, right? So at the end, we are all here to learn, correct? So I hope I'm able to motivate you. And if you have any problem, feel free to drop a mail. You know, in the introductory slide, I have my website link there. You can always drop me a mail. I always respond. And yeah, so as always, I have a question for you guys to ponder upon, which I'm not gonna uh, discuss. So what causes neuronal depolarization? Actually, I was about to discuss this when I was talking about action potential. So there are phases of action potential. There's polarization, hyperpolarization, depolarization. But due to that people were not aware of these things, I couldn't go completely into it, because then I have to start from a very scratch and basic, and the time was not permitting it. So it's all right. If you go to the lecture, you might be able to get more information in the coming lectures. Also, these topics will be covered. Don't worry about this. But try it. If you have little bit idea about what polarization, depolarization is, try to think about this question for the coming week, and I hope that you will learn something new. So thank you so much, guys. If you have any question or want to ask anything, feel free to ask. Otherwise, you all are free to go. Yeah. Welcome, everyone. Welcome. I'm here for 5 more minutes. So if you guys want to ask anything, go ahead. Otherwise, I'm available for next 5 minutes. How many of you have registered for the certificate for this particular course? Just raise your hand. Or planning to. I guess the deadline extended. I'm not aware of those things. But I hope that you guys are registered for the certification. Well, if there are no questions, I guess I'll take leave from you guys. Thank you so much for being patient, and I hope that we meet again next Saturday, same time. Thank you.